Hello students, we'll now discuss about archibacterial cell wall. So in the previous session, we had learned about the eubacterial cell wall that is basically made up of two sugars, an acetyl glucosamine and an acetyl uramic acid. However, in case of archibacteria, one of the sugar is same, that is an acetyl glucosamine, but the other sugar is different. Instead of an acetyl uramic acid, we have an acetyl tallose aminouronic acid, in short known as NAT. So we'll look at the structure of the two sugars. So here is a linear structure of the glucose, cyclic structure of the glucose, and the cyclic structure of an acetyl glucosamine residue, as we had discussed in the previous session. So in case of the second sugar, that is NAD, the backbone is made up of another sugar molecule. It is not derivative of the glucose, rather it is derivative of tallose sugar. So here we can see the cyclic structure of tallose sugar. Further, addition of N-acetyl group at carbon number 2 results in formation of tallose N-acetyl tallosamine sugar. Further, the carbon number 6 is oxidized. And so instead of CH2OH group here, we have presence of COOH group as represented here in the cyclic structure of the sugar NAT and acetyl tallosamine uronic acid. In case of N-acetyl uramic acid, if you remember, we had presence of a lactic acid side chain attached at carbon number 3. We do not show or have presence of the lactic acid side chain in case of N-acetyl tallosamine uronic acid sugar. Instead, the amino acid side chain is directly attached to the carbon number 6, as you can see. Also, as you can notice, all the amino acids that are present in case of archibacterial cell wall, they have a L orientation. Further, the bond between the individual sugars, that is N-acetyl glucosamine and N-acetyl tallosamine uronic acid, is not beta 1,4 glycosidic bond, rather it is beta 1,3 glycosidic bond formed by bond formation between carbon number 1 of the first sugar and carbon number 3 of the second sugar. Here again, the crosslinks are formed between the amino acid side chains that will help in conversion of the linear chains of pseudopeptidoglycan into a complete sheet. Speaking about sensitivity to enzyme lysozyme, so the molecule doesn't contain beta 1,4 glycosidic bond, rather they contain beta 1,3 glycosidic bond, and hence they are insensitive or resistant to enzyme lysozyme. Similarly, the peptide crosslinks are also different here in case of pseudopeptidoglycan, and they are also resistant to amino acid, to antibiotic penicillin. So, in summary, the cell wall material in case of archibacteria, which is known as pseudomurulin or pseudopeptidoglycan, the cell wall of certain archibacteria may contain molecules that are remarkably similar to peptidoglycan or a polysaccharide, that is known as pseudomurin. Backbone of pseudomurin is made up of alternating N-acetyl glucosamine and N-acetyltanosamine uronic acid residues. The molecules differ from peptidoglycan in that the glycosidic bond that is present between the sugars is beta 1,3 instead of 1,4 bond. The amino acids show all presence of L stereoisomers. Cell wall of some bacteria or some other archaebacteria may lack pseudomurine and instead may contain other polysaccharides such as Methylosarcina species have a thick polysaccharide cell wall that is composed of polymers of glucose, glucuronic acid, galactosamine, uronic acid, and acetone. Archaebacteria are naturally resistant to the activity of enzyme lysozyme and antibiotic penicillin agents that either destroy peptidoglycan or can prevent its proper synthesis in case of peptidoglycan or eubacterial cell wall. Thank you. So kindly like, share and subscribe if you like the contents and to get direct updates of any new video that is posted.